served as a teaching assistant and today will present his thesis work. Also, I learned that he worked as a vet assistant both in Turkey and the US before starting his master's here. So you've got lots of interests. And while here, in addition to serving as a TA, he received grants from the biology department, CBEST, and the grad school. His committee is Dr. Bauer, Dr. Jackman, Dr. Langley. Please join me in welcoming Mustafa.
I gotta say, I totally forgot about my resume, including the vet tech part. And fun fact about it is that when I first started working as a vet tech, I was afraid of dead birds. And, you know, vet clinics are not a great place if you don't want to see dead birds. So I got over it through exposure therapy, let's call it. Thanks so much, Dennis, for the introduction. Again, my name is Mustafa. For those who don't know me, I came to Villanova back in 2022, August, and although it's been over 3+ years, I was gone for a good reason. I started working at Oxford Nanopore because of my thesis work that I'm going to be presenting today, actually. And at work, I focus on next generation DNA sequencing, which is something that I'm going to be talking about in chapter 1. Although this is chapter 1, I'm actually going to be start talking about from the chapter 2, 3, and 1. The reason why I want to do it a little unorthodox is because I wanted these chapters to reflect the actual chronological timeline of my master's work, because I actually started with the chapter 2 and then went on to chapter 3 and eventually ended up with chapter 1. But before I dive into any of these chapters, I want to actually talk about what combines all of these chapters together. And first of all, the location. We're going to be mostly talking about New Caledonia or nearby islands like Australia and New Zealand. Second of all, I'm going to be mostly talking about geckos, especially from the family Dipodactylids, and of course, DNA sequencing. But before I go into the details, here's how it all started. In my undergrad years, I helped other scientists working with snakes, skinks, frogs, turtles, and all of those things. And in my summer times, I would go and help scientists with sea turtle research, where I would dig up some nests, count the number of eggs, then do conservation work in the name of trying to understand what is called TSD phenomenon, where the temperature of the nest determines the sex, the gender of the hatchlings, either male or as female. So when I came to Villanova, I had the same thing in my mind, where I was like, hey, can I do the exact same thing with geckos? Because geckos also has temperature-dependent sex determination phenomenon. So I came to Aaron and I was like, hey, can I set up some stuff? And he was like, that's a great idea if you wanted to do a PhD, because that might take years. So instead, why don't you go with something more approachable and something that you can actually finish in 2 to 3 years? And that's how I got into population genetics. And I gotta admit, at the time I didn't know that if I actually tried to study temperature-dependent sex determination, the genetic basis of it, I would probably fail, because there are so many layers, like the epigenetics, the RNA, the DNA. It could be anything, basically. And population genetics, at first, is way more approachable to someone who doesn't know anything about the genetics. And here, we have an example, for example, from California, with the population genetics. If you don't know what it is, what you're looking at is basically either a species or a genus, or maybe even a family, and you try to understand the interactions between different populations, either because they show different morphological traits, or you find something genetically. So this was the entire thing. I'm like, okay, let's do some population genetics. And a couple of days later, I went to him and I was like, can we study this from Sri Lanka, or can we study this from Africa, and can we study this? And eventually we ended up with a genus from New Caledonia. Now, New Caledonia, when we need to talk about it, it is about 1,000 kilometers, which is roughly 700 miles east of Australia. It's very much isolated, and we believe that it was completely submerged and came back to the surface around 35 to 50 million years ago. So all of the species that we're going to be seeing in New Caledonia actually migrated about 35 to 50 million years ago from nearby islands. But why New Caledonia and no other islands? Well, the answer is actually pretty simple. There's an article here from 1985 from Dr. Bauer, and this tells us that he has been the go-to person for any kind of New Caledonia work for quite some decades. And with that, there's a lot of samples available. So that was one of the main reasons why we decided to go with New Caledonia, because we had tons of samples. Second thing is New Caledonia has quite a bit of endemic species. When you look at the species of plants, there are over 1,000 endemic plant species, or when you look at the percentage, over 70% of the plants are endemic to the island. And to give you a reference, the island size is actually smaller than the state of New Jersey. On top of that, the island not only shows endemicism, but it shows microendemicism, which is even a more concentrated version of endemics, not only living in the island, but living in a very tiny portion of the island and cannot be found anywhere else in the island. But we're not really interested in the plants today, we're interested in reptiles. So what do we see with the reptiles is that over 90% of the terrestrial squamates, like we don't really have snakes over there, but skinks and geckos, they are endemic as well. Also, I don't know why this is green, but I guess we'll just go with it. And one of those species, or one of those genus, is Bavaya. So back in 2022, Dr. Bauer, Dr. Jackman, and Ross Sadlier got together, and they were able to publish years-long study where the number of species for the genus Bavaya went from 12 to 41. And it was a huge study with over 200 pages long. And in that study, they also published a huge phylogenetic tree based on mitochondrial DNA. And when you look at that phylogenetic tree, there were a couple of species that showed indications of more work needed. Some further work was needed. One of them is Bavaya gaetana. What do we see here on the mitochondrial DNA tree is that it looks like there are 4 maybe genetically isolated subpopulations of Bavaya gaetana here. A similar thing could also be observed for Bavaya subtuclavis, where we see 4 branches just like this. So the question was, if we look at the nuclear DNA, some of the nuclear genes, would we also see these 4 branches happening, the same topology basically? So the road plan was pretty clear, but before that I want to point out something. In the most taxonomical studies, especially these days, you have to include 1 mitochondrial gene and 1 nuclear gene. Now the reason behind that can be complicated, so I'm not going to actually dive deeper into this, but I'll just tell you that the 2022 paper didn't have any nuclear genes included in it. They only published the mitochondrial DNA. So that was the next step. I was going to sequence some of the species for their nuclear DNAs. But there's a big problem with most nuclear DNA, is that their rate of evolution is pretty small compared to the mitochondrial DNA. So we need to use rapidly evolving gene groups, but which ones? At that point, we're going to be looking at a paper from 2020 where Dr. Jackman, Dr. Gamble, and Dr. Karen got together and they published about 180 genes of nuclear DNA where they are rapidly evolving. So they could be a good substitute for mitochondrial DNA where you're not allowed to use mitochondrial DNA, or sometimes you're just looking for extra data. And one of those genes is the PKDREJ gene. It has high variability, it has a long coding sequence, and it is present in almost all amniotes. With the high variability, it was going to give us a better chance studying not only the species level, but also subspecies level interactions, and this is what we're looking at. And on top of that, when I first started, I focused on the A copy of this gene, but I realized that there's also the C copy, the second copy. B copy is missing from the geckos, and I was like, I'm already doing the A copy, so why I shouldn't throw in the C copy as well? There's going to be more data, and it could be useful. And that's what I did. Again, I'm going to go back to the 2 of the species that I wanted to focus on, Bavaya gaetana and Bavaya subtuclavis. What is important about them is that they both habitate the southern portion of the island. And I put two maps over here. On the right side, we have the geological map of New Caledonia, and on the left side, we have the vegetation map of New Caledonia. When we look at these both maps, I actually kind of want to focus on the left side here a little bit more because it shows small islands of forests that is covered by some other type of vegetation. This tells us that there are small chunks of island that may be isolated from the rest of the islands, either because of the geological differences or because some are sclerophyll forests and some are mudkey-like shrubs. So this might be the effect why we are seeing that some of the species in the southern portion of the island are isolated from each other genetically. But is it actually? We'll see. So we're going to go back to the 2022 paper, and what we're going to do is, as I mentioned, we have 4 branches, and we're going to grab some samples from here, 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 and put them up on the geographical map to see if we actually see the isolation, as we mentioned. And what do we find? What we see is that there looks like a little bit of isolation or clustering happening. Like the brown ones are usually closer to the northern part of the island. The teal one is kind of on the very southern part. There's the purple one. And then only in here near Mount Kogi we have 2 subpopulations getting close to each other. And we're going to get back to that point. But as of now, I'm just focusing on, looks like they are clustering completely isolated from each other. But is that actually the case? For Bovia subtuclavis, we're also looking at the same thing. Let's grab some representatives and let's put them up on the map. Kind of like Bovia gaetana, we're also seeing that maybe the orange ones are closer to the northern portion of the island and everything else is kind of on the southern. So maybe there is a little bit of clustering happening as well, but we're going to test these out by using the nuclear genes. So now that I have my hypotheses set, I could focus on the wet lab part, where I followed the very traditional, very accepted approach. I designed some PCR primers, then I did Sanger sequencing, and after that, with that data, I made some phylogenetic trees, and eventually I did population genetics analysis. So let's take a look at the results first. This is the Bovia subtuclavis tree, and unlike the mitochondrial DNA, where it was clustering in 4 subclades, here we don't really see 4, and maybe, if you really dig into it, we might be seeing 2 clades. But is that actually the case? Well, phylogenetic trees do not really do a good job telling us the interaction between the subpopulations. So in this case, we need population level genetics analysis. So with this said, we're going to be looking at one of those examples, which is the structure analysis. When you look at this, there are a couple of simple things that's going to explain what are we looking at here. For example, each of these bars represents one of the samples that we're looking at. Orange is representative of one of the subpopulations, and blue is representative of some of the other populations. You can think of it as orange are golden retrievers and blues are doodles, let's say. And we have some samples that only carries the orange genes, and we have some samples that carries only the blue genes. The 2 things important here that I want you to look at is the first one, we're looking at the locality of these samples and see if they're also clustering with what we see here. And when we look at the Goro Plateau and Plan de Lac, it seems like the blue samples are all from there, but we also see fully orange samples or some hybrid samples from the same locality as well. That tells us that there's not enough isolation. The second one is that we have hybrid samples at different levels of hybridization. That tells us something, that it's not, the hybridization didn't happen just once, but it is a continuous ongoing process that has happened in the past and maybe still happening. So this tells us enough that these are not isolated populations. And when you reflect them on the geographical map with the structure data, now it looks like here the hybrid ones are kind of clustering with each other, but when you look at both the structure analysis and also we run a mental test where we look at take the structure analysis and try to correlate that with the geographical distances between them, it basically does not support the idea of there are two subpopulations here and they are isolated from each other. Instead, for Bavai subclavis, we're looking at one continuous population that may be interacting with each other at different levels. But when we look at the Bavaya gaetana, again, this is the tree that I got. We are kind of looking at different topology as well. It's not 4 clades. Instead, we're looking at 3 clades, maybe 3, maybe 2. There's the orange one, there's the blue one, and there's this 2 of the samples that is clustered by themselves. And when we look at the structure analysis for the Bavaya gaetana, we see a totally different story. What do we see? We see orange samples that are completely isolated from the blue samples, with only 2 samples being the exception. Now we'll get back to those 2 samples. But when we reflect them on the map here, this was what we were looking at, the mitochondrial DNA. This is what we find out. The blue ones are all pretty much isolated from each other, isolated from the rest of the orange population, except one locality, and that is Mount Kogi. And those samples from Mount Kogi, they are these 2 starred ones. So what I found out in Mount Kogi was that for the Bavaya gaetana, there are 2 subpopulations that are almost completely isolated from each other throughout the island, except one single locality, and that is Mount Kogi. So when we found out this, the question was like, okay, apparently there's something funky going on in Mount Kogi, so why not we take a look, a deeper look into this? And that is what we did. And that's going to be the next chapter. We're going to be looking at more Bavaya gaetana samples at that point, not only from Mount Kogi, but also from the rest of the island as well. We're going to be sequencing their ND2 gene and also their nuclear genes, because some of the new samples do not even have anything yet. We're basically pulling them out of the freezers. And also we're going to be including Bavaya savaggi. Why? Because Bavaya savaggi lives sympatrically with the Bavaya gaetana on Mount Kogi. So we're like, okay, let's throw in some Bavaya savaggi as well. For the mitochondrial DNA results, just like the Bauer study in 2022, this is what we find out. The topology is kind of similar. We see 4 different clades formed with the mitochondrial DNA, but also some of the new samples that I sequenced with mitochondrial DNA clustered sister to the rest of the Bavaya gaetana here. So we're like, is it a new species? What's going on here? But at the same time, I had nuclear DNA as well. So the question was like, okay, let's take a look at the nuclear DNA as well and see if we're going to see separation from the rest of the Bavaya gaetana as well. 2 things that I want to point out here is that 1, just like the topology turned from 4 branches to 2 branches with the nuclear DNA, although we added even more Bavaya gaetana species at this point, it still clusters as 2 subclades. And the second one is the putative new species. It does not cluster with the rest of the Bavaya gaetana as a sister group, but now it clusters with the clade 1 and 2, which clusters that as a sister group to the rest of the Bavaya gaetana. So that tells us that although on a mitochondrial DNA scale, some of the species are isolated enough from the rest of the Bavaya gaetana, when you look at the nuclear DNA results, that does not support that hypothesis. So maybe in a couple of more million years, we might be something happening, but as of now, we cannot call them isolated new species. And the other thing with the nuclear DNA is that we have some Bavaya gaetana samples at the top, which clustered with Bavaya savaggi from Mount Kogi. So not only we found hybridization at Mount Kogi for these samples, that they were hybridizing, they were the bridge between the 2 subpopulations of Bavaya gaetana, but they are also hybridizing or introgressing, that's the term, with a totally different species, which is not something you see every day. So that was pretty cool to find. And when you reflect these results on the geographical map, the putative new species cluster all together at the northern portion of the island. But again, we cannot call them new species because of 2 different factors. 1, nuclear DNA the nuclear genes does not support the idea of them being a new species. And second of all, we work with a scholar, Ross Sadlier. He looked at them on a physiological level, and he couldn't find any traits that separated them from the rest of the specimens that you see here. So those were the important parts. And this is how this chapter ends. And at the time, I was about to apply to PhD programs, and so I went to Aaron and I was like, "Hey, Aaron, can I apply? What do you think of this?" He looked at my resume, and I didn't really have a strong resume, and he was like, "If you want to get into a competitive school, do you maybe want to extend to a third year where you can actually focus on not classes anymore, but more research?" So I was like, "Okay, I can do that." So I wanted to do it. And at the same time, Todd came to me and he's like, "Take a look at this paper. Look what they did. Do you want to maybe do the same protocol as that?" This is a paper from 2023 from Ben Karen. And what we see here is the mitochondrial DNA of a skink. What they did was pretty simple. They did 2 very large PCRs to cover the entire mitochondrial genome of these skinks, and they were able to grab the complete mitochondrial genome by running those 2 very long PCRs. So at that point, I had almost 2 years of experience with Sanger sequencing, and I felt comfortable enough. Okay, let's try this. I knew how to troubleshoot PCRs and everything, and I thought it was going to be a walk in the park. And if you've never run a PCR before, this is what a successful PCR usually looks like. You have bands that are aligned well, and every sample has bands. So when I started running the protocol, I was expecting a similar result, but it just did not happen. Either I found bands everywhere where I was not actually looking at the stuff, like 500 base pair fragments, 1000 base pair, or I basically didn't even grab anything. And it just kept happening again and again and again and again and again. And the issue with PCR is that it can be a pain in the butt to troubleshoot failed PCRs because there are so many factors. It's almost like cooking. What is too much here? Is it the extension length? Is it the extension temperature? Is it the annealing temperature? Is it the polymerase quality? Is there contamination? I don't know, maybe I'm not running the gel electrophoresis right. There's so many things happening, and that makes it even more difficult. And on top of that, this PCR protocol is a long PCR protocol where it takes 10 to 12 hours to do 1 PCR. So at best, I could run 1 PCR per day and maybe 4 to 5 PCRs every week. And that means I had to stay in the lab pretty much all the time. But that was the only way I knew, so I'm like, okay, let's do it. How did I do troubleshooting? I started from the very basics, like changing the water source to sterile water, to anything like changing the annealing temperature, the extension length, to perfect primer design. I actually, for some samples, sequenced their 16S DNA, sequenced their ND4 DNA, put them up on the Genius, and designed primers based on the perfect DNA sequences. So technically, or theoretically, the primers should fit them perfectly, but it just didn't happen. And after about 6 months of trying, lots of time, lots of money was basically wasted or spent. Todd came to me. It was, I think, May of 2023 or 2024, and he was like, we're wasting a little too much money here. Maybe we should give up on this. And I was like, okay, it is what it is. But I was still curious why this was failing. So it was at the time that we were going to attend some conferences. And at every conference, I started asking around. I'm like, hey, I'm coming across this issue. What would you do? What would you do? To the different scholars, to my former professors. And someone suggested that maybe I should change the way we do the DNA extractions. And never have I even thought about that. Had I even thought about that up until that point. But when I came back, I had just a little bit of that Taq polymerase left, that special polymerase left. And I was like, before I use it all up, maybe I should just change the DNA extraction method. And at the time, we were mostly using a kit-based DNA extraction method called QIAGEN, but there's also other ones from Thermo Fisher, Zymo, and I was like, okay, maybe I should do something else. And this was the protocol that we would follow. And I switched it up to salt-based extraction. Now, salt-based extraction is a relatively old method and requires a bit more babysitting, but it has two critical differences between the QIAGEN method and the salt-based extraction method. The first one is that QIAGEN kit has these spin columns, where once you put the liquid in, you centrifuge it, so everything has to go through that spin column. And the second thing is the amount of pipetting. When you look at the numbers, for QIAGEN kits, you need about 5 rounds of centrifuging and about 6 to 7 rounds of pipetting. For the salt-based extraction, you need about 2 rounds of centrifuging and 3 rounds of pipetting. And when you look at some of the articles, like this one from the 2011, the number of pipetting affects the fraction of intact DNA very heavily, very quickly. Just from 2 to 4, it almost goes down to like 50% of the fragments are now chunked into half. And on top of that, the level of vortexing or the time of vortexing also affects that. And not only that, but a lot of the samples we have used, they have been sitting in the freezers for maybe 10 years, 20 years, maybe even 30 years. Or they were going through freezing and thawing, so they were out of the freezer and back in the freezer, out in the freezer, back in the freezer. So that also affects the fraction of intact DNA. Or even if they were sitting in the perfect condition, like minus 20 Celsius, which is what we do in the freezers here, the DNA is still not that stable, and over time it can still degrade. And they even show that, like, theoretically, it can even keep degrading at minus 268 Celsius degrees. And the important thing about this protocol is that you have to have the DNA fragments that are larger than 10,000 base pairs to be able to amplify anything. Otherwise, this protocol is going to fail automatically. So what I did, I changed the protocol to salt extraction. And once I changed the DNA extraction method, everything changed. Now, out of the 20 samples here, only 1 failed. Now, some may still fail, because primers are not perfect, they're not going to bind into everything perfectly, but out of the 20, 19 is great. And when we look at the larger results, what do we see here? For the first primer set, that is sequencing about 10,000 base pairs, and on the second one, that's sequencing about 10,000 base pairs. Out of the 24, only 1 failed, the first 23 samples, sorry. And out of the first 23, 4 additional ones failed for the primers. Now, this is a great find. And out of the total 36 samples I had, 3 samples failed for the first primer set, and an additional 6 failed for the second primer set. Now, why did they fail? Again, it might be the not perfect primer design, or maybe the DNA has degraded under 10,000 base pairs, so it cannot amplify anything anymore, because, again, we need larger fragments. And I was not going in blind. I always do DNA extractions, and after that, I try to see if there's DNA extracted with about 1,000 base pair primer sets. But again, just because they didn't fail with 1,000 base pair fragments doesn't guarantee that they're going to work for 10,000 base pairs. But they already gave us a very good baseline. For the first primer set, we're looking at over 95% success rate. And for the second one, we're looking at around 83% success rate. And the first one is pretty much universal for this family. And the second one works for 4 out of 5 samples, which is still great because you're getting the entire mitochondrial genome with this protocol. So this part, up until this point, I tried this primer on all of the diplodactylids from Australia, New Caledonia, New Zealand, and they kind of like kept working really well. We tried them on different families of geckos, and they failed, so that was a good indicator that these primers are universal primers for that family, Diplodactylae. So we kind of found good primers if we wanted to do long PCRs. But just this, so far, is the materials and methods part of the study. But we can also look at the analysis part. So here on the top left, I kind of want to focus on this part first a little bit. We have a typical reptilian mitochondrial DNA order. Now I'm saying typical, but it might not be so typical. What I want to focus on here is the control region and the 2 tRNA before that. Most of the time, people have been focusing on the other part of the mitochondrial DNA, either from 12S to the cytochrome B genes, because they're relatively easy to design primers in the conservative parts, and then you get a lot of data after that. And the control region has been ignored, or the tRNAs has been ignored very heavily, because especially the control region is a very dynamic part of the mitochondrial DNA, where designing primers is very difficult because of two main reasons. One is that there's so many repetition going on. There are so many repetitive coding sequences within the control region, or the primer design is basically almost impossible with Sanger sequencing. There are two important things that I want to focus on here. The first one is for short read next-gen sequencing methods, or whole genome shotgun sequencing, where they fragment the DNA into 150 base pair long chunks, and then they assemble them on the computer. And the second one is the Sanger sequencing. With the Sanger sequencing, you can get up to 1,500 base pair chunks, usually around 1,000 base pairs. So if your control region or the repetitive parts of the control region are anything longer than 150 base pairs, you are automatically eliminating the possibility of being able to use shotgun-based whole genome or Illumina-based next-generation sequencing. And if your control region repetitives are going over the 1,000 base pair line, you are eliminating the possibility of Sanger sequencing. And what we found out for all these samples here, although I'm showing 6 of them, for some reason, for the 24 samples that I was successfully able to assemble, all of them showed coding sequences that were repetitive. 9 of them had coding sequences small enough, the repetitive part small enough that would be able to capture with either Sanger sequencing or Illumina-based next-gen sequencing. But the other 15, they showed different results, where you have to use either Oxford Nanopore sequencing or you have to use PacBio sequencing to be able to grab the actual control region. So that was the important find. The second one was control region fragmentation. Out of the 6 samples we look at here, not the typical one, we all saw some sort of fragmentation in their control region. Now, when we look at here, we can actually see the fragmentation. This is broken into 2 pieces instead of just 1 piece now. This is also broken into 2 pieces. And this is broken into several pieces. These are short enough that the programs are not able to, annotate, but we still see small chunks of what is that kind of regions that we're looking at in the control region or near the control region. The second thing was duplication. Exception being this sample, all of the other 5 ones we see here showed proof of duplication. What do I mean with the duplication? Is that this part, when you copy it, it can not only be found here, but it can also be found within this chunk as well. Or this can be found here. Or when you look at this part, the control region is almost all completely identical with this part, and they also can be found here as well. So it's basically just duplicating itself, and that's what's creating that dynamic environment where people were not able to capture. And lastly, we're looking at the tRNAs. Pretty much all of the samples, except being this one here, showed duplications of tRNA. Either the set got duplicated completely, or just the protein got duplicated, or the threonine also got duplicated. For this one, it doesn't look like it got duplicated, but instead it just moved from one location to the other location. And I'm pretty sure if we had a larger data set, and if we actually analyze this further, these findings, either the duplication, the fragmentation, the tRNA findings, or even the coding repeats, these can be used as a phylogenetic marker, just like what people have been using for the cytochrome B gene or the 12S gene. But for now, let's just look at the fragmentation, the duplication, and the coding sequence. When we reflect our findings on on the tree here, this is the phylogenetic tree of family Diplodactylids. The first thing I want to look at is the repeating coding sequence patterns are present. Again, just like I mentioned, all the samples that I was able to assemble, they showed repeating coding sequence patterns. The second one is if there's control region fragmentation absent or if it's present. When we look at the New Zealand geckos, the exception being Torupuku, the control region fragmentation was present. So their control region fragmented into at least 2 pieces. For the control region fragmentation absent, New Caledonia didn't show any fragmentation except being one single Bavaya species. And I sequenced 9 or 10 species from Bavaya, and only one actually showed fragmentation. So that's probably a small evolution that happened on its own, like a random thing. And for Australia, I was able to only get 2 specimens successfully assembled. The reason behind that is I actually want to try many more, but we didn't have a lot of samples from Australia, and the ones that we had, the DNA was basically just not still intact anymore. I just tried doing regular DNA extractions with them, and even Sanger sequencing for them has failed for 1,000 base pairs. So that was not pretty much possible to do anything. If we had more samples from the family Diplodactylidae from New Zealand or Australia or even New Caledonia, we would be able to make many more speculations or actually come up with real results and can tell, oh, maybe this is happening and that is happening. Or on the phylogenetic level, you could say control region fragmentation happened, I don't know, 50 million years ago, before the parent of New Zealand geckos moved to New Zealand from the family Diplodactylidae. But with the results I have as of now, it is pretty much impossible to come up with conclusions like this. But regardless, even when we look at such small thing, we can kind of, on the surface level, see that control region, once you get the whole mitochondrial genome, might be used for phylogenetic, tree binary data sets. So, with that being said, that was the references. Come on, where do you go? These are all the references I included in the presentation, and more can be found in the written copy of my thesis. And I'm ready for any questions.
At this stage, it's the general audience.
That's true that you showed. Was that made using the control region sequences, or was that made from other mitochondrial genes in like an intentional state reconstruction?
This was made with mitochondrial genes.
Okay.
Yeah, Matthew Feineke.
And then you mapped on the control.
Yeah, I just mapped it off. I took the entire mitochondrial tree. I put these icons or the shapes of the islands, and then I added the stars or the squares based on if I found anything or not. It was basically a very similar binary yes or no questions, basically. Almost like you look at the physiological traits of an animal.
With the Bavaya septiclavis, with the different subgroups that were going on in that species, I think one of them, like they had their distributions had different sizes. Do you know if there's like a reason that, like one of those subgroups had like a way bigger distribution than all of the other 3, which seemed to be more
endemic. Let's bring the map up for this one, I guess. This is the Bavaya septiclavis. We'll look at the orange specimens.
Yeah.
And you're asking why the orange one it has a larger distribution area compared to the rest of them?
Yeah.
Well, there could be many factors. Bavaya septiclavis is a terrestrial animal. So it's actually way more dynamic. You can find it. And maybe if it had more habitat areas, it could have been spread even further. But there's no particular reason. Well, the question actually can be said this way: why all of these samples can only be found in this like small area compared to the rest, while they can actually spread a lot further? Because with the Bovie septiclavis, they could actually spread further, but for this southern portion of the island, they just wanted to inhabitate that southern portion of the island. And again, many reasons can be said for that. Maybe the temperatures are better there, or they want to stay a little higher in the elevation or lower in the elevation because the elevation is higher over here compared to the rest. So yeah.
So they seem to be, like, those 4 subclades seemed like decently divergent in their mitochondrial genomes. But obviously, especially that those southern populations are all introgressed in the nuclear genome. Do you have any hypotheses for why those subpopulations have maintained distinct mitochondrial haplotypes but are introgressed across the nuclear genome?
So, for that, again, I'm just speculating. This might be the early stages of a speciation. Because mitochondrial DNA evolves way quicker than the nuclear DNA, what we would be maybe seeing here is that these populations have been isolated from each other for 500,000 years, let's say, and they are not genetically interacting with each other. So that's why on the mitochondrial genome, which evolves very rapidly, almost 50 times faster than a regular nuclear gene, we're seeing the proof of that, but it hasn't passed enough time to reflect the same results on the nuclear genes.
Okay, so you think these things completely isolated by barriers, or do you think there's contemporary introgression going? Like
For Bovie Gaetana,
yeah.
I'm assuming they are pretty much isolated from each other,
Okay.
exception being the Mount Kogi.
Okay, gotcha.
Because it's not just the mitochondrial DNA, but also the nuclear DNA is pretty much showing a similar result as well. Mantle test is showing it supports the hypothesis. Again, we can't really say that for sure, but maybe give this a couple more million years, maybe 2-3 million more years, and they might even become their own separate species if they do not keep interacting with each other at Mount Kogi.
How does human impact this island in terms of moving populations around?
That's something that I was wondering as well, like demonic intrusion. That was a question. I'm like, how do we know? Because this island is very heavily mined. So that was a question I asked Aaron like 2-3 years ago. How do we know that a huge mining truck did not take a huge piece of land and moved one of the species from here to there? But he was like, that usually does not happen. That was his answer.
Or was it?
Yeah, maybe happening but usually the forests part of the island, unlike the coastal parts, it's not very heavily populated, so we don't see a lot of anthropological effects happening.
How are you ruling out incomplete lineage sorting in this potential hybrid? How are you ruling out incomplete lineage sorting in this potential hybrid area? Because hybridization is not something being passed down from older generation.
Good question. I don't think I ever thought about that yet. Complete lineage sorting. Could be, yeah.
I have another question.
Yeah. Do you want to speculate?
This is unrelated. Well, why did you land on PKD or EJ for your gene as opposed to all the other relic genes to choose from?
I guess I don't really have to go back to this. For certain species, we already had a small database. Bovie robusta, I think that was one of the species that had their genes available for that study. And when I started looking at the variability rate, PKD-REJ was coming at like the number 6 or something like that. It was very high, and also the coding sequence was over 2,000 base pairs, which kind of makes it easy to design any kind of primers. And the PKDRUJ primers were designed by Todd. Then I designed the C primers as well. And it could have been something else as well, but it was just up there, very high top. So I'm like, okay, let's go with this.
We had, so who did we have to? Oh, I should.
Could have been like NXRA5 as well,
Yeah, I know. But from whose data was that from? Remember?
Shannon?
Shannon.
Yeah, Shannon, yeah. So, could have been something else as well, but definitely I wanted to use it. It was one of the ones that I wanted to use it because it had a high variability rate. And it worked well. Like, you try different primers, and when you find some of them that actually pretty much works for all of the samples, you just keep going with it.
Now that we've broken protocol, at this point, you all are welcome to stay, or if you need to leave, feel free to leave, and we will go to the committee, whoever would like to go first.
I have to go, but good job.
Thank you. So you showed a map at the beginning of the geology of New Caledonia.
Yeah, I'm going to do this.
And it sounded like you were going to link that to these population structures, because they did seem, well, you showed two maps and they had a lot of parallels.
It's this one, right?
There you go.
Uh-huh.
Okay, so this is the vegetation
The
composition.
vegetation map on the left side, and geographical
Which
map.
also is reflected in your population structures.
Mm-hmm.
So what's the driving factor here?
So my take is for, let's say, Bovia gaetana or Bovia subtuclavis. The vegetation map highly reflects the results of the geological map here, either because of the soil structure or some other factors. It creates small islands, the isolated islands. And I think, let's say, Bovia gaetana only likes to live in the maquis-like shrubs, or Bovia subtuclavis, let's say, only likes to live in the maquis-like shrubs, and Bovia gaetana only likes to live in the evergreen humid forests. They can only be found here, 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 here,
Okay.
and vice versa. I think that's what's causing the isolation. Maybe they got separated at some point, or they prefer different habitats. And
How do they get to Mount Kogi?
Mount Kogi,
Kogi. I wrote
which
down
is
the
Mount Kogi is right here,
Oh, okay. I was thinking it was further.
so it's not too far from their distribution area
Yeah.
for either of them.
Because it seemed like, if you had more thorough sampling along the contact zone, maybe you would see more hybridization and introgression.
Maybe. I showed 2 samples hybridizing
Yeah.
there, but there were actually 6 samples,
Okay.
if not more, that I sequenced from Mount Kogi. 2 of them showed,
Okay.
and the other ones, like in this map, we can actually look at it here. Like we can see one fully blue one. So although this is on Mount Kogi, that is not interacting with the other population. So maybe only certain lineages are interacting with the other populations.
Okay. I'll ask some genetic method questions, which I don't normally do.
Sure.
But so the Karen paper, what extraction method did they use?
They actually did not said a lot about their extraction method.
Yeah.
They just said we just did like some salt extraction, but they didn't really focus on any of the stuff that might be contributing to that. And technically, the extraction method might not be the main factor for their stuff if they are using fresh tissues.
Fresh samples.
Yeah, if you're using fresh samples, fresh tissues, and if they haven't been sitting in the freezers for so long, even one copy that is large enough is enough to amplify, and then after that you can keep working it. So maybe they haven't even come across any issues, and that's why they never talked about the DNA extraction method in the protocols.
Okay.
I emailed them, and it was not even something that we discussed. He
Right.
never thought that DNA extraction would be the main
Right.
factor.
Okay.
Mhm.
And these repetitions in the mitochondrial DNA, are they exact, like to the base pair repetitions? And
You have chunks of like 150 base pairs that has 11 copies.
identical?
Identical. Or you have 75 base pairs that has 7 copies.
Okay.
Or sometimes within that 2,300 base pairs, you have 2 different coding sequences that can be found, like one can be found 6 times and the other one can be found 11 times. Varies in range. It could be as small as 50 base pairs, repeating itself up to 600 base pairs, or it can be something that's 250 base pairs that's just repeating itself to.
So if
500.
you had a 250 base pair sequence and you had one base pair different, would that tell you that it was a repeat? I mean, can you distinguish it then?
You could, but I actually didn't find any.
Okay.
It
So
was
they're identical.
basically fitting perfectly. Boom, boom,
Wow,
boom, almost
alright.
like a Lego piece. It was just making that stare super easily.
Well, that's annoying.
Yeah. Actually, once you get the coding sequences, just put them up on the Word or text edit and Control F, copy and paste whatever you're looking for, and it shows up as yellow highlighted all over the place. And that's how you can tell. And just in between, I put an asterisk. I'm like, okay, 1 copy, 2 copy, 3 copy. It's super easy to find out. You don't even need to go on to and write a pipeline code for that,
Right.
which I would do if it was necessary, but it was like so blatant up there.
Thank you.
I'll go next. Okay, so let me take you through a thought experiment. You
Sure.
suggested that the control region might be good for making phylogenetic trees, but
Okay.
what we're just talking about, what happens if you had 8 250 base pair segments are all identical, and then you line them up and you try to make a tree out of it? How good would your tree be?
What I would do in that case, I would probably approach it almost like the Huntington's disease. I would look at how many repeating base pairs. At that point, it's not a binary thing of yes or no. At that point, it's like how many repeating 250 base pairs I have, and maybe that could give it. Just like some of the physiological traits, how many scales do you see on the temporal region?
Right, but the other thing is the repeat segments are identical, are often identical or close to identical within an individual. But if you compare 2 genera, do those repeats even exist often? The exact same repeat?
I wouldn't think so.
Right.
Yeah.
So that means that trying to line them up.
Yeah, it would have to be
Right.
subspecies level.
So that I mean, I think that those things explain why people generally don't use the control region to make phylogenetic trees because of those
But the thing is,
issues, right? So,
yeah,
I mean, except for right in the actual place where it's actually acting as the control region. What does the control region do? You should know this. Like, what function does the control region have? Do you know?
people didn't really find a lot of think about the control region.
So it is the origin of replication for the mitochondria,
Oh,
right? So it does have a function. So, and it's in that area where it's actually functioning as not where the repeats are, but where it's actually there. For very close relatives, you can use it because it's the most variable part of the entire mitochondrion. That's what's used for humans, right? For example, like when we can. That's probably the best place to link together human populations on a scale of thousands of years, right? So, but once you get to different genera, you can't even compare them.
I actually wanted to include many more samples when it came to the analysis part, because 24 samples is not a lot. But the issue was when you go on NCBI, you can find complete genes of 16S, 12S, or even if they share the mitochondrial genome, they refrain from using the word complete mitochondrial genome because they know that it might not be complete because maybe they don't have the entire control region. And I maybe found 1 or 2 samples that was actually complete mitochondrial genome on NCBI, and I was like, okay, I cannot use this. So neither the preliminary thing or nor at the end of the analysis, I couldn't actually look at it from a higher perspective,
Okay.
a larger.
Totally different question, right? So when you think about Guytana and Septuiclavus and you sort of look, when you see there's an area in southern New Caledonia with lots of variation,
Yeah.
right? An area north of that where there's less variation.
Mm-hmm.
What does that generally? Do you know what interpretation do people have when they've got it within a single species? You've got lots of variation versus an area with a lot less variation.
Yeah.
What would be the normal interpretation for that in any species?
In any species.
Right, an area with lots of variation versus an area with less variation, right? In terms of time,
I would say microendemicism. That's the first thing I would say. So maybe there's like a small portion of the island, because that's kind of like what I asked Aaron back in the day. I'm like, why do we see like these are too close to each other, but why are they isolated? And it could be the elevation, a very quick, rapid elevation change, almost like a valley. And these are like finger-sized animals.
Well, I'll tell you what I'm thinking. It should be that those are the oldest places, right? So the older population is in the same place as a large population, the more variation you get.
They
right?
went to
So-
a place and then they came back, kind of.
Well, no, just that area is the ancestral area.
Mhm.
That's probably the most likely explanation for both Subsuaclavus and for Guytana, and that they moved northward.
If I had to speculate, I'd probably think that maybe Mount Kogi, for the Bavaya Guytana, could be the origin. Then they spread into the southern portion and the northern portion of the island. But for maybe Subsuaclavus, that's where they originated first. That was something that we discussed previously. But it could be, again, several different factors. But origin of species could be one of them.
See if you remember this. What does PKDREJ do?
Oh,
What's its function?
sure. It is the, yeah. So people actually looked at this with the humans. It started as, back in the day, in the human genome, they were trying to find something that would inhibit the function of sperm, so they could use that gene and just block it, and maybe find the perfect contraceptive for the human genome, so if we could. But once they were able to disable the PKD-REJ gene, they realized that it would maybe delay the fertilization by 2 hours, but it wouldn't stop it. What's it called? The EJ part is the egg jelly part, and it is about the acrosome reaction, if I remember correctly. But that's pretty much what's the function of it.
Okay, so you do remember.
Yeah.
All right.
Yeah, I love that story of like how did that become a thing, and then could have been a good story about, oh look, we found the perfect contraceptive method where it's not, damaging to either of the genders.
Can you go back to the two maps together, the
Sure thing.
vegetation and the geological map?
Yep, 2010,
Yep, here you
2008
go.
paper.
So on the geological map,
Mhm.
the green is what?
Peridotite. So high nickel, high metal, any kind of heavy metal containing soils.
What's another term for that that's used in New Caledonia? The soils are characterized as
I had that in my thesis proposal. I don't remember it top of my head.
ultramafic.
Yeah, ultramafic. Because Bavai ultramafic.
Okay.
Yeah.
If you go to well, I forget which map. Go to the map that has Mount Kogi on it.
It's the one that's zoomed in to Mount Kogi.
Yeah. Okay.
Not the perfect shape.
What's weird about the fact that Mount Kogi, not that Mount Kogi has both forms, but that Mount Kogi has any of the forms?
Could you elaborate the question?
So there's something unexpected about even finding the species at all at Mount Kogi. What does every other locality have in common that Mount Kogi doesn't have?
I don't know the answer to that question.
Mount Kogi is not ultramafic.
Oh.
Mount Kogi is the one non-ultramafic, and pretty much when you look at Bavaya across the island, the species that are on ultramafics are only on ultramafics. So this is an exceptional case that this peripheral population here, right at the edge of the distribution, is in the one place that, every other dot is smack dab in an ultramafic area, going to be all covered with maquis. There's no maquis on Mount Kogi, none. So Mount Kogi is sort of a weird in that way. So any ideas why it might be on Mount Kogi at all, and why you would get both things on Mount Kogi? And I'm not looking for anything specific here.
I'm looking at the speculation at that point. If the samples that we collected from Mount Kogi, if they are flexible enough to live in a completely different habitat, that's why they would be there, and maybe that what makes them able to interact with the other subpopulation as well. So maybe it created a totally different lineage, or maybe it may be creating a totally different lineage.
Okay, as I said, there's no, I'm not looking for any
just
particular. So, what's a possible geographical, topographic feature that separates the two populations from one another there?
So this part we're looking at.
Yeah, why are the orange one? What might account for their separation?
So although when we look at here, the first thing that comes to mind is that there's a river, but that's actually a very new formation. It's not something that accounts for it. I would probably say, as the name calls out, this is the Goro Plateau. It is almost like a valley, a huge valley where the elevation goes down very drastically. So maybe these samples just cannot climb up anymore. Or the second thing would be the elevation. This elevation it may look like it is also elevated here, but these samples are surrounded by an elevation change. That's what makes them keep there. That's what keeps them there, so they cannot actually interact with the blue population anymore. I haven't done the field.
I would say that that there is a river there, and you correctly pointed out that the lake that you see there,
Rivière Bleu, yeah.
Right, that's a new lake. That's a man-made lake, but that's the Rivière Bleu system that runs through there, and that might be a subdivision. If you go to your other close-up map for Septuagint,
Like here.
yeah.
Yeah, this is the isolated part where
Right. So
that is the Plan de Lac.
there you have a couple of things going on, because you were talking about the Goro Plateau. So the Goro Plateau is essentially on the far right side of your screen. And then the Plaine de Lac, those lakes that you see down there, those are natural lakes. Those are the only natural lakes in New Caledonia.
Yeah.
So, one of the things that might be going on is your populations there that are mixed, they're actually on the Goro Plateau, but all the other ones are in the Plaine de Lac, not on the plateau.
Yeah.
So, when you look at this, it could be that the geography is playing out differently in the two species. That in one, it doesn't really matter, Plaine de Lac versus Goro Plateau.
Yeah.
But here, maybe it does make a difference.
Yeah.
Something to think about there for this. So, if you could sample someplace else, maybe zoom out a little bit on this,
Yeah,
so
sure.
you have the whole distribution on there. If you were to sample someplace else, in either species, you have your choice. If you Gaetana or Septuagint, and you could sample some area that we don't have samples from,
Yeah.
where would you sample from?
Where would I
Do
sample
you think
from?
there are any unsampled localities that we just don't have material from, or is there some place where you would?
Let's go on the
Come.
Bavaria Gaetana map as well. What I would do, I kind of know the answer to, but I would probably get closer to this region as well. We don't have a lot of samples to the left side. For Bavaria Gaetana, it's kind of important because that's where the orange population is getting closer to Mount Kogi, and we don't have samples from that region. And for Bavaria subtuclavis, it's kind of a similar story as well, where we don't really have a lot of samples from this region. So maybe, like, there's a separation that's happening actually starting from here, and there's a small valley that we don't see. Right? So I'll probably focus on a little bit this part of the island.
If you go up on the, just kind of, that's fine. On both maps, on both maps, just above where your cursor is, and to the right, above and to the right, you have that area that doesn't have anything in it. Do you know why there's nothing in it? Why there's no localities in there?
Because that's where the river flows into the ocean? Or?
Well, I mean, the river comes out at the one point there, but that whole sort of area.
A huge elevation change? Or what's causing it?
No roads.
No roads?
No roads. That's why there's no
like human-made roads?
There's
or?
no samples because there are no roads.
Oh.
It's hard to get into. So, you know, this is something to remember in a place like this as well. That the pattern that you see
untouched
is not necessarily what the real. You know, you're seeing some of the pattern, but we don't really know what's happening in some places. And the northern Gaetana, if you go back to the, sorry to make you flip back and forth between maps, but the
Pewter New Species map. Yeah, this is closer to the coast, so it's more accessible.
And so those are new localities. They weren't originally included because those are relatively new. Like we didn't have those in 1990 or in 2000. We didn't have those localities. So there's still so many unknowns. We're not really sure. You know, so you know this. I think there's room for growth here in the sense that you could take a look and make some predictions about what you might find in different areas, and then you know
more samples from different
try
localities.
you know see what happens when we get more samples. And I had one question about you were talking about the in your the final part in chapter 1, you were talking about things that didn't work for some of the taxa. So if you go just to the tree, it shows where the things don't work. That's fine. So some of these things certainly make sense why they didn't work, but I'm wondering about Strophuris, because we have relatively new big series of Strophuris from I don't know, let's say 2018 or something. It shouldn't have been anything wrong with that. So did you? I mean, that I can't imagine that would have had any integrity problems to the tissues themselves.
Yeah, I think I grabbed 2 Strophuris, and they were back-to-back label numbers, so maybe it was not the perfect primer set for them. And them being the consecutive label numbers is not always a good thing, because that means that they might be coming from a very similar locality, which means that their genetics are going to be very similar as well. So maybe if I tried more Strophuris from different label numbers and different localities and different field trips, I could have been more successful. But Australia, I really, really wanted to grab more samples from.
And finally, based on this tree, if you could have one genus that you don't have, or something that didn't work, in terms of understanding the evolution of this fragmentation and
the key one,
what one would you pick?
Probably based on the historical thing, I would probably go for Crenadactylus from Australia, because that would give me almost like a baseline for, just like I mentioned, for the New Zealand. It looks like they all changed from the rest of the group here 50 million years ago. That tells us a change, almost like the geckos lost the B copy of their PKDREJ. That's a complete change, or it's almost like a genome duplication happened. And so if I could grab data from Crenadactylus, that would probably say, oh, for all of these specimens, there were actually control region fragmentation was present, but for New Caledonia and Australia, they actually lost those copies, so it would probably be very useful.
Yeah, I think Crenadactylus is the way to go. And I guess I had one last one. You looked at diplodactylids, and you were saying, okay, you'd like to have more samples. in reality, the sampling you have is enough to be interesting, right? I mean, I think that's a publishable result to kind of show that there's a pattern, and there's at least a hint of a pattern that in New Zealand something else has happened. What do we know about the fragmentation and repeats in other Geckotan families?
To be honest, we don't know much because there isn't a lot of studies that focused on the control region necessarily, because again, historically it's super hard. They tried, like the Komazawa, even in early 2006 ones, they tried nested PCRs where they fragmented the mitochondrial gene into 6 pieces, but it was very labor intensive. So a lot of people just refrain from focusing on the control, the region, or the complete mitochondrial genome, because when you look at the amount of time you're spending to get the amount of data, focusing on cytochrome B, focusing on ND2, focusing on 16S is way more valuable. This was something that I was able to get because I wasn't necessarily trying to focus on the control region, but it was almost like a side effect of what I was doing. So I was able to get, but we don't really know much about the other geckos. That is, we don't have a large data set to compare them.
Good job.
Thank you.